morning students, welcome to B.Sc-II Zoology class. Today we are going to study the human development up to implantation. Human development up to implantation. So first let us see the fertilization. Where fertilization occurs in human beings. So this is the uterus, female reproductive system, uterus, fallopian tube, ovary. So during ovulation, ovum is released from one of the ovaries, and soon after the release of ovum, the fimbriate of one of the fallopian tube picks it up and takes this ovum into its cavity. So these are fimbriate, finger-like projections. They capture the released ovum. Now the ovum has entered into the cavity of the fallopian tube, and this is surrounded by the follicular cells. And the egg which is released from the ovary is in secondary oocyte stage. Clear? Secondary oocyte stage of second meiotic division. Okay, so its development is stopped at second meiotic cell division. Metaphase of the second meiotic cell division during the oogenesis. At that time, at this stage, secondary oocyte stage, the ovum is released from the ovary. That ovum is taken into the fallopian tube, and this is here isthmus ampulla. Here, when it comes here, the sperms enter. At that time, if coitus occur, intercourse occurs, the sperms are released just nearby the cervical opening. From the cervix, they enter into the uterus. From uterus, very few of the sperms they enter the fallopian tube. Here, the fertilization takes place. What is happening? Fertilization. Fusion of male and female gametes occur. Soon after fertilization, the secondary oocyte completes its second meiotic cell division. As a result, first polar body is released. And now this cell is having haploid number of chromosomes. Egg has haploid number of chromosomes, and sperm gives another set of chromosomes. These two sets of chromosomes are mixed, and this is called as amphimixis or karyogamy. Karyogamy means fusion of nuclei. Male pronucleus, female pronucleus, both are fused. This fusion is called as amphimixis or karyogamy. After the fusion of these two, the number of chromosomes becomes 46. Two sets, 23 plus 23, 46 chromosomes are established. They are present in the egg. Now, after the fusion of these two gametes, egg becomes zygote. Why this is called as zygote? Zygote has diploid number of chromosomes, whereas the egg has single set of chromosomes, haploid number of chromosomes. So this is zygote. So zygote undergoes, soon after the formation of zygote, this zygote is a single cell stage. This undergoes cleavage. See, after fertilization, embryogenesis starts. What is embryogenesis? Development of embryo from fertilized egg up to the formation of young one. Right? So during the embryogenesis, several changes occur that leads to the formation of young one. Right? So that is called embryogenesis or embryo development. embryogenesis. So embryogenesis completes into three stages: cleavage, gastrulation, and organogenesis. Cleavage, gastrulation, and organogenesis. These are the three stages of embryogenesis. Embryogenesis, during the development of embryo from fertilized egg, these changes are occurring. The first stage is cleavage. What is cleavage? Cleavage is a series of rapid mitotic divisions to give rise to multicellular embryonic stage, wherein the embryo doesn't show growth. After the division of cells won't grow in size. Their size doesn't undergo increase or increase in the embryonic size. Size in the embryo doesn't increase during the cleavage. Only cells number is increasing. The single cell zygote becomes multicellular. Smaller cells are forming. As the number of cells increase, size of the cells become decreasing. Right? It is decreasing. So as a result, what happens? But the multiplication of DNA is very rapid. Multiplication or synthesis of DNA is very rapid. Okay? So this is called cleavage. So first cleavage in human cell, human embryogenesis, the first cleavage is meridional. First cleavage occurs along the animal core vesical pole axis. This is called as meridional cleavage. At the end of cleavage, two cells are formed. These two cells are called blastomeres. Therefore, the first cleavage is holoblastic and unequal cleavage occurs. These two cells are not exactly identical in their size. One is larger, another is smaller, right? Unequal sized. So, still it is externally covered by zona pellucida layer is still there. It is there because of the presence of zona pellucida. These cells are intact. They are present inside the zona pellucida. Okay, blastomeres. So first cleavage occurs 30 hours after the fertilization. After 30 hours, the first cleavage completes. At the end of first cleavage, two cells are formed. These two cells are called blastomeres. One of them is larger, another is smaller. Next cleavage, second cleavage. So second cleavage is right angles to the first one. Right? So first smaller cell gets divided, later on larger cell. Thus, at the end of second cleavage, four cells are formed. Four blastomeres. Still, these are extremely called by zona pellucida. So the second cleavage is right angles to the first one, or latitudinal. So the second cleavage is completed after 20 to 30 hours of first cleavage. After the formation of first cleavage, second cleavage completion takes 20 to 30 hours time. Okay? Next, the third cleavage. Third cleavage is occurring at different planes. Irregular planes of cleavage occur. This is 8-cell stage. This is still externally covered by zona pellucida. The third cleavage completes in 12 hours after second cleavage. So, as the womb is moving towards the uterus, zygote is moving towards the uterus, cleavage is occurring. This is single zygote, it is 2-cell stage, then 4-cell stage, 8-cell stage. So 8-cell stage or 16-cell stage is is known as morula stage. This is called as morula stage. It is a solid ball-like structure. It has several cells. Later on, what happens, the size of the peripheral cells increasing. Just they increase. As a result, the size of the morula is increasing. Three times more than the what you call zygote. As a result, pressure is created inside the zona pellucida. So as it comes here, the uterine wall. So uterine wall has two membranes. Three membranes actually. This is endometrium, myometrium, and perimetrium. Okay, perimetrium is there. So this endometrium, by this time, goes on secreting, nutrient secretions, and these secretions are absorbed by the peripheral cells. As a result, pressure is created inside. Now the zona pellucida is ruptured. And inside the embryo, cavity is formed, and this cavity is filled with fluid secreted by the surrounding cells, peripheral cells. These peripheral cells are known as trophoblast cells. These are called trophoblast cells. Now what happens, when it comes here in the cavity of the uterus, as the size of the embryo increases, the external covering, zona pellucida, gets ruptured. Now the embryo comes out of the zona pellucida, and the embryo is surrounded by trophoblast cells, larger and flattened cells. These are called trophoblast cells. They are phagocytic. They release enzymes, lytic enzymes, and these enzymes, what they do, they erosion of the endometrium occurs, so that the embryo gets embedded, attached to the endometrium. The attachment of the endometrium by the blastocyst is known as implantation. Blastocyst. See, after the release of zona pellucida, development of the blastocoele. This is called blastocoele. This is called blastula stage, and this blastula attaches to the endometrium. The attachment of the blastocyst to the endometrium is known as implantation. The process of implantation in human beings begins 7th day after the fertilization. 7th day of fertilization, it begins, and process of implantation is completed in next 3 days, right? So as long as the embryo is surrounded by the zona pellucida, the embryo doesn't attach to the endometrium. So soon after the release of zona pellucida, the embryo becomes naked, and now it can attach to the surface of the endometrium. This attachment of the blastocyst to the endometrium is known as implantation. So the process of implantation involves many stages, many steps. Okay, development of trophoblast cells, then chorionic villi enter into the endometrium pits, right? Like that. Later on, the entire blastocyst gets embedded into the endometrium. The entire blastocyst is immersed into the endometrium. The surface of the blastocyst is covered by endometrium. Thus, the process of the implantation is completed in 3 days, right? So after the implantation, relationship between the mother and the embryo begins. nutrients that are coming from the mother, blood circulation, they are passed on to the fetus through the placenta. Okay, we'll see it, how the implantation begins, how the implantation ends, okay, in the next video. So these are the steps that are occurring in human beings after fertilization. So reproductive organs, the process of oogenesis, spermatogenesis, all you have studied in PUC second year. So the process of implantation is important here. Cleavage and implantation in human beings. Okay, in the next class we shall see how implantation begins. Thank you.